So are you guys, everybody good with the PowerPoint slide? Just at least one or two people, if they say yes, that will be great. Yes, or anything?
Yes.
Okay, perfect. Yeah, thanks, Cooper. All right. So we always usually look at the DNA replication in terms of cellular context. And so your question could be that where is the viral DNA replication? Because that many of the DNA viruses are also the part of your Baltimore scheme that we talked about. The reason we are doing the viral transcription, and you know that, anyone, why we are doing the viral transcription immediately after the RNA, after studying the RNA viruses, RNA synthesis? Anyone can chime in? The reason or the rationale behind it that we are doing the transcription after that, which is making of the mRNA from the DNA template. Yeah, Matthew, that is correct, because it involves RNA synthesis. So that's exactly what it is, because you remember that the thing that the essence of the Baltimore scheme that we talked about is essentially the synthesis of the mRNA. That's actually the first event that actually happens once the virus actually goes into the cell, and that's basically we are going to talk about. So in the last time, what we did, if you see that the bottom of the slide right here, that we talked about the RNA viruses, where we talked about the mRNA synthesis, that is the first thing, and also we talked about their genome RNA synthesis. Now here, we have to separate these out into two other phenomena. What we are going to do that right here, if you just look at these viruses under the red box, and they are all highlighted, these viruses do have a DNA in their reproductive cycle. Now, make sure that you very clearly understand these viruses right here, adenovirus, hepatitis, parvovirus, they all do have DNAs, either double-stranded, gapped, plus, minus, but the retrovirus actually do not have a DNA genome, but it does have the DNA in their life cycle, and all are going to and they're all going to do the transcription. So why we are using the word transcription here, because this is exactly the similar process of the cellular phenomena where a DNA template is used to make the mRNA. So you can see a double-stranded DNA template is used to make the mRNA, and this is what we define as a transcription. We tried not to use the word transcription in the previous chapter with the RNA viruses because they use the RNA template, and we use the word mRNA synthesis instead, which is sometimes we just cannot make the differences, and we have to use it, but we try to avoid use that. So this is what we are going to use it, and you can see that what we are going to see that these are the polyoma papilloma virus, SD40, very famous of them. Probably the most worked on virus is SV40, simian virus 40. Then you have the herpes virus, large genome, adenovirus, mediumly large genome, hepatitis B virus. This is probably the one which is discovered, the newest one in there with a gapped DNA, very unusual. And you have the retroviruses. There are now retroviruses, when we are talking about this, just only one family. Remember that your HIVs are the retroviruses. There are feline leukemia viruses, the retroviruses, but there are many other retroviruses as well, which are not as complex of HIV. They are very simple genome, but still they are retroviruses, means they infect the host, they will integrate their genome, they will convert their genome to the reverse transcriptase into the DNAs, and that DNA will be integrated into the host chromosome. So that part is always in there. So that's what we're going to talk about. So here, what we are going to say that although we said that mRNA synthesis or the transcription is the first event that is going to happen, when the cells are infected with the DNA viruses, at least one protein, or often many, are needed for the DNA. So this is hold on one second. Yeah, that's fine. So at least one protein is needed for the DNA replication. Now, the idea is that what we are not talking about the transcription. At least one protein is needed for the DNA replication. So what we need to do that replication, we need at least one protein. And without mRNA transcription, viral proteins can't be synthesized, but not all DNA templates are ready for transcription. This is the main issue. And we are going to see that how they can be ready. But some can be ready, which is an example right here in the SV40. So let's take a look at just the part of the life cycle that once the genome is delivered into the nucleus, if you carefully look at the blow up of this genome, what structure is this? Can you tell from what it is? This is how the chromosomes are packed in our eukaryotic genome in the chromosomes. So this just shows you, anyone? No? This is a nucleosome, which you have these octamers of the histones. You have that H2A, 2B, 3, and 4, each with 2 copies. And this is the octamer core of the histone, and you have 140 base pair of DNA around it. So this is a typical structure that how the eukaryotic genome is in the chromosome. And polyoma genome is exactly like that. And that's the reason that this is actually ready for the transcription, and it will immediately make the mRNA, and the mRNA will synthesize the protein. But now what you need is that even in this, the DNA replication, which is right here is the DNA, this portion is the transcription, DNA to DNA making is the replication, will need at least one protein for the replication of the DNA. And that is your LT, or the large T antigen, that what we are going to see. And also what we are going to see that it will not only also require for the DNA replication, the LT, or this large T antigen, we are going to talk about that, also will be responsible for the second round of mRNA synthesis. So it will be a transcription factor as well. So the same, at least one protein, this is the simplest one, is responsible for the DNA replication or onset of the DNA replication, as well as a second round of mRNA synthesis, which will mainly make the structural proteins of the virus. See that what first happened immediately after the genome gets into the nucleus of the cell, the mRNA is synthesized, and that is making this LT, which stands for large T antigen, ST for the small T antigen, and we are going to talk about that in detail. These are the regulatory protein, only LT function has been shown. These are the regulatory proteins which are transported back into the nucleus and have an effect on the replication, as well as the second round of mRNA synthesis. And we are going to temporarily divide these parts as the stages of mRNA synthesis or transcription. And we will talk about these as immediate, early, and then late synthesis, which is not shown here, late synthesis of mRNA or the transcription. Here you can only see a early and a late, but there could be immediate, early, then late. Many complexities will arise. So that's basically the overview of that, why this first transcription is needed, at least one protein needed for the DNA replication, and also it can help in the second round of transcription. So here are some viral DNA genomes, and that must be converted into templates. And you can see that the hepatinovirus, which is the gap genome with a RNA attached to one of the 5' end, and then you have a protein attached to the 3' end. It has to be repaired into a double-stranded genome. And we are going to talk about these much detail in the next chapter, not this one. On the other hand, the parvovirus have this very weird type of structures where you can see that these two sides, which is the T-shaped structures, actually contain the ITRs, or the inverted terminal repeats. And here is you have an open 3' hydroxyl, and from here the strand is synthesized all the way through, and this will be the double-stranded version of the DNA, which is ready for transcription. Retroviruses, you have a plus strand mRNA. first, the viral reverse transcriptase will do the reverse transcription, make from this plus minus intermediate, which is not shown. On that minus intermediate DNA strand, the plus will be formed. Now you have a double-stranded DNA. Now the integrase, also a viral enzyme, will integrate this double-stranded DNA into the chromosome. Now, chromosome of the host. So now this part of the retrovirus has become a part of a chromosome somewhere in the host, and the transcription of this double-stranded DNA will continue. So what you can see that all of them need a double-stranded template before the transcription can begin. Right here, you can see this. And most of them are not ready in that form, and this is how they can be ready. All right, so now we are going to take a look at that. What are the cellular or eukaryotic DNA-dependent RNA polymerases are present to do this function? Now, remember that we talked about this RDRP all the time, is RNA-dependent RNA polymerase, the hallmark of the RNA viruses, because you have an RNA template. But here, what we are going to talk about, the transcription is a eukaryotic phenomena, where you have the DNA template, and on top of that, you are synthesizing the RNAs. And there are several eukaryotic RNA polymerases which actually can carry out this function. We're going to see that in case of some very large DNA viruses, they code their own RNA polymerases as well, but we'll come back to that. But the majority of the DNA viruses, or the viruses which have DNA somewhere in their life cycle, right, the retroviruses, will be using these enzymes, or the eukaryotic enzymes, and all of these are present only in the nucleus, not in the cytoplasm. So the synthesis of all of these are going to happen inside the nucleus. And it's a de novo synthesis, no primer is needed. The RNA polymerases bind to the specific site and start the initiation. And you can see that the RNA pol 1 in the cellular, they make the pre-mRNA. Whether it actually transcribes any viral RNA or not, it is not known. RNA pol 2 is the one that we are going to discuss the most, and this is the enzyme which actually make the pre-mRNA and also make the primary microRNA and small nuclear RNA. But this pre-mRNA is the main thing, which is the unspliced version of the mRNA, or let's not use the word unspliced at this point. We'll come to that. That is correct. But let's say that this pre-mRNA is not ribosome ready. This pre-mRNA must have to be processed to a mature mRNA and exported out of the nucleus when it will become ribosome ready. So the pre-mRNA is still not ribosome ready. This is also used by many viruses to make the pre-mRNA for the viruses as well, which will be processed to ribosome-ready mature mRNAs. Also, just remember that this is a common mistake that actually happened to a couple of people, that when you write miRNA right here, that is microRNA. And this have a silencing function. It just binds to it's different from the siRNA. So remember that these microRNAs are just small, in between 18 to 25 nucleotide long, single-stranded, and they will bind to the cognate mRNA sequence to silence them. And they are very, very specific as well. So this is miRNA and mRNA, very different. Read very carefully in the exam. It will not be any trick question, but sometimes it can trick you. And also the RNA pol do the small nuclear RNA. And what you are seeing that here is only one known exception. This hepatitis delta virus is a satellite, and this satellite does have a viral RNA genome. And the RNA pol 2 somehow replicate. This is the only example where a cellular, now remember that very carefully, it's a cellular DNA-dependent RNA polymerase. So transcription is done by the DNA template. Only in the HDV genome, which is an RNA genome, this RNA pol can recognize the RNA as a template and can synthesize its RNA. And this is an extremely unique, and it is the only exception where the RNA pol 2 shows this RNA-dependent RNA function due to some of the secondary structures that is formed by this HDV RNA genome? So this is a very good question, and definitely remember that. And then RNA pol 3 actually do make the transfer RNAs. It also makes the 5S ribosomal RNAs, which are the component of the ribosomes, and also the U6 small nuclear RNAs. These are like 6 uridines, U, like that. And these are the functions for the RNA pol 3. And these are also used by some of the viruses like adenovirus virally associated RNAs. We will mention that. And then you have this Epstein-Barr virus, EBER, Epstein-Barr virus encoding RNAs. Then this is a murine herpes gamma virus, primary microRNA. These are some of the examples that is used, that these viruses use the cellular RNA pol 3 for making of these RNAs. So on the other hand, just make sure that you understand, and we're going to talk about that. Adenovirus, when we're going to talk about, all its mRNA is going to be synthesized by the RNA pol 2. But this is virally associated RNAs are not mRNAs. They have different function. We are going to see that in a different chapter. That is going to be synthesized by the RNA pol 3. So this gives you an overview of that. And again, this gives you an overview of that and what we are going to talk about in this chapter and in this topic, and the next topic is just depicted here. So what we are going to talk about is just the top line here, which is what you are seeing, that the plus minus DNA, double-stranded DNA, and this red arrow is designated as the transcription start site. That's what it is. This is where the transcription start. Now see that the transcription start site does not mean that it is at the 5' end of the gene. There are some sequences which is upstream of the transcription site or initiation site that is required for the machinery of the transcription to bind in here. Often we call it a regulatory sequence. Or transcription can start anywhere in there. Sometimes transcription can start in the other direction as well, and they are usually denoted by these arrows. And the rest of the process, which is the capping, polyionization, then nuclear transport and splicing, these are the modification of the mRNA, and we are going to talk that in the part 2 of this topic. So let's take a look at the detail about the transcription. The first thing is that transcription is highly regulated. It must be regulated. So think about not virus for a second. For human beings, eukaryotic, we have approximately about 25,000 genes, 26,000 genes. At each time in one type of cell, if you take an epithelial cell, say any epithelial cell, probably just about in between 3,000 to 5,000 genes are expressed in that cell. So basically 20,000 genes are silenced, but that nucleus of the epithelial cells contain all the DNA for that 20,000, those 25,000 genes. You take a neuron, or you take a cell, any cell type that you take at one point of time in any kind of animal will only express just only a fraction of very few number of genes that is required for that type of the cell. Now, there are two differences of that, and this is not needed, but just to understand that some of the cells will have about 2K to 3K proteins, which are, we call as a housekeeping genes. Every cell have to have that. Say, for an example, if we just are talking about any of these, say a sodium-potassium pump. A sodium-potassium pump has to be in each and every cell on the body has to have that. That's a housekeeping gene. A glucose transporter. Glucose is needed for each and every cell to produce ATP. That is needed. So there is some kind of glucose transporter, not GLUT1 or GLUT2 or anything like that. There are many different types, but at least one type of glucose transporter needed to be on one of the cell. But there are many which are very specific. Say, for an example, the neuron produces the neurotransmitters. So you have all the enzymes required for the synthetic machinery to produce the neurotransmitters. These genes, which are responsible for making neurotransmitters in a nerve cell, are absent in all other cells throughout the body. A liver cell, heart muscle cell, regular muscle cells, skeletal muscle cells, or epithelial cells will not have that. So you can easily understand that transcription is highly regulated and it is very, very cell specific. So how this high degree of regulation is achieved is shown here. And this is basically a transcriptional control region. In reality, it is much more complex. So let's take a look at that, what happens. So what you are seeing right here, that this +1 means that this is the start site, and this is usually the first codon that will go into the mRNA. Usually that's the start site. And you have the initiator sequence right here along with that. Now remember that AUG is a start codon for mRNA. And that AUG, the first start codon, is the signal for the ribosome to make the first methionine residue. It has nothing to do with this. This is entirely different. We are not talking about the AUG, which is a start codon. Around that start codon, although which could be here, you have something else or a different sequence in the DNA, not on the mRNA, on the DNA, which we call as an initiator sequence. And this sequence, part of it right here, if it is downstream of the AUG, will be the part of the mRNA as well. So this initiator sequence is okay for the RNA polymerase to bind and synthesize the mRNA, but usually in an extremely low rate. And if anybody want to recap that the lac operon, if you remember that when you do not have lactose in the environment, the E. coli do the expression of these lac-related genes in a leaky fashion, like just a few copies. That is the same thing, that without any of these controls, sometimes the RNA polymerase can make a couple of transcripts just by starting with the initiator sequence. But it is absolutely very, very slow and almost not of any functional use except a few transcripts. But this defines the specific accurate start site. Now you have this the famous sequence right here, which has the repeats of these TATAs, and we call this as a TATA sequence or the TATA box. And this binds to a transcription factor, and remember this name. This is your transcription factor 2D, which is a TATA binding protein, and this is one of the very famous one. And then you can see that there are some other regulatory sequence upstream of the TATA sequence. And let's define the word. Suppose this is the gene start site, +1. If we are going right here towards the 5', which will be, say, 0, -1, -2, like this, this will be the upstream of the gene. If we are going from the +1 start site towards the 3' end, that will be the downstream of the gene. So just make sure that you define the upstream and the downstream of the gene and what it means. So now you can see that there could be local regulatory sequences upstream of this core promoter. So the core promoter is where the RNA polymerase and the associated protein actually binds primarily, which is right here, the TATA sequence and the initiator sequence. Upstream of that, which is just a couple of 100 base pair away from the TATA sequence upstream, are the local regulatory sequence, and other proteins can bind in there, also can initiate transcription from these sequences as well. On the other hand, what you can have distant regulatory sequences, and the proteins that bind in these distant regulatory sequence are usually called the enhancers and silencers. Now here it has been shown as upstream, but remember that these enhancer or silencer sequence could be downstream of the gene as well. Now, you can easily see that if the proteins are binding in these regions, that may have an interaction with these RNA polymerase and the associated subunits. This is easily understandable. They are within a couple of 100 base pair. If this is 10,000 base pair away, how they are going to control the transcription from this region, that we are going to see the next. So right here, what we are going to see, that just another rendition of the generalized transcription regulator site. These are the sequences of transcriptional control in some of the other examples. And you can see that it could be varied. So what you are seeing right here, this is the initiator sequence. And you can see that that's the start site. This is your Tata box. And these CBF and USF, and you do not need to remember individual names of these proteins, just to have an idea that these are your local regulatory sequences. Whereas SV40, you can see that there is no specific initiator sequence, but there are multiple start sites for different genes, and you have the Tata sequence, and there are several repeats of these DNA sequences which are called which binds to this SP1 protein. So this is not SPL or special or anything like that. This is called the SP1. This is one. This is like a Roman 1, SP1 binding site. So this is local regulatory sequence. On the other hand, you can see that this is the same adenovirus. This is the portion which is then which actually transcribe early genes of the adenovirus, where you have the Tata sequence, and then you have an E2F binding site, and this is actually the important one that we are going to talk about. And this is a transcription factor binding site, and we will talk about that, how this controls the regulation of the early genes as well. And you can see that almost all of them are within the 100 base pair region, and we call this one as a local regulatory sequence and come in all different shapes and sizes. Now, what we are going to see is what happens to these distant regulatory sequences. So if you look at this, that what we talked about just now, right here, this example, and the local regulatory sequence, that's in the top. So if you see that the two DNA elements and you have the two elements, think about that. This is just for an example, that this is a Tata sequence and this is a local regulatory sequence. And these are the transcription factor which are actually binding in between them. And this could be RNA polymerase as well. And then one subunit of RNA polymerase, and then they can interact closely. When this happens within a few hundred base pair, we call this as a cis-acting element. So this is a cis-acting element, this is a cis-acting element. But if they are 10,000 base pair away, this type of interaction cannot happen, and the DNA must loop, and this is called the DNA looping model of the transactivation. So here you can see that this element is right here. This element is on the opposite side, and that's why we call it in trans, just opposite. Cis on the same side, trans on the opposite side, and they can interact with this protein to enhance or silence or repress the transcription as well. So here you can see that this is an example of the SV40 and how complex it is, and do not have to remember all the names of them, and we're just going to talk about just a couple of them. So you can see that initiation complex, you have the RNA pol2, and these are the different subunits, and you can see that this is the transcription factor 2D that is famous. Also remember that the transcription factor 2H, we are going to talk about that as well, and that is in the C-terminal end of the RNA pol, responsible for phosphorylation and some specific functions. We're going to talk about that. These two are the ones what you can remember: TF2D and TF2H, and these are all assembled over the local regulatory sequence, and you can see that this is the start site. On the other hand, you can see that this enhancer binding is almost 10,000 base pair away, and the DNA loops, and these are the DNA binding proteins which can enhance the transcription of this initiation complex. Just the take-home message. The process is much more complex, and we do not need to go into that detail. So what are the overall, is there any general idea about the proteins which regulate the transcription? So they are host or virus sequence specific DNA binding proteins. It has to be, because remember that no matter what, all of these proteins has to bind on the DNA to exert some of its effect. And the viral coactivating molecules do not bind to the DNA, but can modulate the transcription, are also required. And we will actually see some of these coactivating molecules which will bind to some of the DNA binding proteins on the other hand, not specifically directly to the DNA, that modulate transcription. And many coactivators also modulate the structural activity of nucleosomal templates, histone methylation or acetylation. Here, one of the important things that just concept, remember that when you actually do methylate or acetylate the histones, it actually, what it happens that it will wrap around the DNAs around the nucleosome very tightly. And as they are tightly packed, the transcription factor will not be able to bind on the regulatory sequence and recruit the RNA polymerases to initiate the transcription. But if these methylation are demethylated and these acetylations are deacylated, and these are done by the HDACs or the histone deacetylases, if they actually deacetylate these histones, at that time, this DNA, which is wrapped around tightly in the nucleosome, will loosen up so that the regulatory sequence are now exposed, transcription factors can bind, and RNA polymerase can synthesize the mRNA. So tightening and loosening up of the DNA does that. And this is what is known as the heterochromatin, that those are the non-translated regions or the tightly packed regions of the DNA, whereas the euchromatins are the transcriptionally active regions of the DNA, which are loosened up DNA, that you can just remember or recap. So the sequence-specific transcriptional activators do have some specific, very specific common features. So one is the DNA binding. And for the DNA binding, and this is, all of these are very important for your quizzes, exams, all of them may have one corresponding question. They will have a zinc finger, which is also basic in nature, which means there are basic amino acids. And then the helix turn helix is also another type of motif that is found in this DNA binding region, which wraps around the DNA as a clamp. And remember that why, so the question is that why the DNA binding domain has to be basic. Anyone? Quick question.
DNA has a negative charge.
Yeah, very, very good. All right. So DNA is acidic. That's why it's a nucleic acid. And DNA have a negative charge. You have to have a positively charged area. Very good. That has to be basic. On the other hand, you will have a dimer formation domain, which actually contains, which is a motif called a leucine zipper. So the leucine zipper actually dimerizes, and there are several examples of this. So what happens that the transcription factors are present in the cell in a monomeric form, but only upon dimerization on the DNA sequence, they will be functional and will recruit the other transcription machinery and the RNA polymerases. And also there are some activation domain, and several different types of protein can bind in there, which could be acidic. Now remember that the proteins could be acidic, and that is not the part which is basic, but they could be acidic, they could be glutamine rich, they could be proline rich, isoleucine rich. There are many different types of protein that can be found to activate the DNA as well. So these are some of the general rules of the sequence-specific transcriptional activators, which recognize very specific sequence in the regulatory region of the transcription. So here we are looking at the strategies of the transcription of the viral DNA. And if we're just looking at the origin of the transcriptional components for the host, this is used only by the retroviruses with the simple genomes. These are very small genomes. And then you have extremely small viruses like colimavirus, which causes your cauliflower mosaic virus. And circovirus is the smallest known virus. And these just have 5 to 7 genes, less than 10. And they are responsible, or the transcription of their RNA from their DNA template, or are only, they only use the host factors as their transcriptional components. So all their transcriptional regulatory domains are bound by the host transcriptional protein. Now, remember that very carefully, what we are trying trying to convey here. So all these viruses goes into the nucleus, or say, for example, the retroviruses will be integrated into the genome as such, and they are now trying to make the mRNA. To make the mRNA, you need the transcriptional control. That means to activate the regions of the DNA so that the RNA polymerases can synthesize the mRNAs. To do this, these transcriptional control proteins are all provided by the host. That is what it is. But here what we are seeing, that these are some of the ones which are the viral protein of the late genes like bacteriophages. They need the host proteins like this, but at least one viral protein. And also you can see that in the herpesvirus, parvovirus, papilloma, polyoma, retroviruses with complex genome, they also need at least one viral protein in addition to the host transcription factor. This one viral protein is the viral transcription factor. So here, this is the host transcription factor only required for the transcription. Here, host transcription factor plus, or some of the host, there are hundreds of transcription factor, some of the host transcription factors, and at least one viral protein. But you can see that right here, these adenoviruses or the herpes viruses require host transcription factor and less than one protein to stimulate the transcription. And very interestingly, see that the adenoviruses have medium-sized genome, herpes virus is fairly large genome. And then what you can see that right here, giant viruses like pox viruses, Epidura viruses, Mimi viruses, all their transcription factors are viral. They do not use any host RNA transcription transcription factors. So you can see that as the genome size, so if you just go from top to the bottom, smallest, medium, a little bit larger, and largest, this is the increase in the genome size. And you can see that as the genome size increase, the number of transcription factor provided by the virus actually increases. So now here is a very important idea. Up to this point, host plus one or host greater than one, whenever you have the host, all of the transcription is going to take place in the nucleus of the cell because host transcription factors are active in the nucleus as only. So they are transported, all of them are synthesized and transported in the nucleus. But this is the very unique thing. As the giant viruses produce all their transcription factor by themselves, these pox viruses and the giant viruses can replicate in the cytoplasm by itself. They also do synthesize their own RNA polymerase. So the whole of the transcription machinery is coded by these giant viruses. The only thing that they cannot do, they can even make some tRNAs as well. The only thing they cannot code is the ribosome. That's why they are still obligatory parasites in that sense. So this is a very important concept. I just wrote it down clearly. So definitely understand it, because you can have some critical thinking question that why these can replicate in the cytoplasm, not in the nucleus. All right. So after that, we are looking at some of the patterns of regulation of the transcription by the viral proteins. So here, very simple to understand. The first one, which you call as an autoregulatory loop, and think about that. This is just a positive or negative feedback loop that you have seen in biological sciences everywhere. So what it is, you have this initiator sequence and you have a gene X that produces the viral mRNA and translated into the protein X. These protein X by itself will bind onto the regulatory sequence of its own, and it can enhance the viral transcription. If it enhances the viral transcription, then we will talk about, we will tell that it is a positive autoregulatory loop. On the other hand, this can inhibit this one as well. If it inhibits its own transcription, we will call it as a negative autoregulatory sequence that we have seen in our system all the time, in a physiological system, negative autoregulatory loop and the positive. We also call these a positive feedback and the negative feedbacks. So on the other hand, there is a cascade regulation, and it is just shown just only with two, but it could be many as well, where the first gene X is transcribed and translated into the protein X. Now the protein X will bind to the transcriptional control unit of the second gene right here. So this is another transcriptional unit. This is transcriptional unit 1. This is transcriptional unit 1, and the regulatory sequences vary. Protein X will bind to that and control the expression of gene Y. Now, this is just only shown in two-step. Now, gene Y can regulate the expression of gene Z as well, and that has been seen as well. So it could be multiple regulation as well. So this is the generalized scheme of the regulation of the transcription of the viral proteins, and we are going to see some specific example after these. So the first one, we are looking at the simian virus 40. This is a polyomavirus, and you can see that this contains the double-stranded DNA, and they are enwrapped just like eukaryotic DNA. So what we are seeing that we have already just give you the overall hint of that. And remember that although it shows the entire life cycle, we will just only consider the one that is needed or relevant in there. Say, for example, the entry we have already talked about, the packaging and all those kind of assembly and the packaging we're going to discuss in a separate topic. But here what is happening, that once the DNA is delivered into the nucleus, as we have said, and this is the repeat, it will produce the early mRNA. That early mRNA produces the early protein, which is LT, is the famous one, large T antigen. That will be transported into the nucleus, and it has two functions. One of them, it will start the DNA replication, and it will start the second round of mRNA synthesis. In the second round of mRNA synthesis, these mRNA will code for the structural proteins, which will assemble, which will be again transported into the nucleus and assembled into the viral particle. So you can easily see the first step, which is the early, synthesize the regulatory proteins, whereas the late phase synthesize the structural proteins. Unless you replicate the genome, there is no need for synthesizing these structural proteins, because without the genome, there is no, you cannot package the anything. Or if you say, for example, what happened, your question could be, what happened that if you produce these proteins at the very first stage along with that, what will happen? Yeah, of course, that can happen as well. And the RNA viruses actually do that. We have not seen this early late phase for the RNA viruses. They just do transcribe all of them at the same time. The only type of regulation that they have, that they make the mRNA amount in different amount. And usually for the regulatory proteins, they make the mRNAs in a little bit less amount. They make mRNAs in a larger amount for the structural protein. So RNA viruses just only have that control, but not this early or the late phase control, what you are seeing right here, and we're going to see in a couple of other examples. The reason being that it just waits for the DNA to be replicated in sufficient amount so that can be encapsulated by the viral structural proteins. So how this is controlled? By the SV40. This is the widely known, and the thing is that one of the things that all these cell biology or the molecular biology, they were all discovered and came into your cell biology textbook from these viral systems as well. So they were almost all discovered in the viral system. You can just easily look at that. And that's probably the reason that I teach cell biology as well, because they were all discovered in the viral system. So what you are seeing, that this is the temporal organization of the SV40 genome. And think about that. These are the early genes, and these are the late genes. So what happens that right here, it's a circular DNA, and this is the origin of replication. Right here is this is the early area, early genes, and right here is the late genes, for example. And this is the linear representation of the same thing. So the first, what happens, that it produces the early mRNA, and this early mRNA will make the LT, which is, we just saw early mRNA making this LT, or the large T antigen. So you can see that large T antigen will do two things. It will enhance the viral DNA synthesis, and it will also enhance the transcription of the late genes that we have seen, which are right here, transcription of the late genes. Right here, this is your enhancement of the DNA replication, and right here, enhancement of the mRNA synthesis. That's what we are actually seeing. So this is the overall scheme, and this is a very, very simple process. Now, this is a very important concept. We are going to see that as the not only the LT directly enhance the late gene expression, viral synthesis also enhance the late gene expression. And we're going to explain that unique phenomena, which we call as an anti-repression. Anti-repression means that escape from repression. So somehow these L region, again, carefully understand that this L region of the DNA is repressed by some factors. When the viral DNA synthesis occurs, like increase in the genomes, more and more copies of this, those repressors are removed by some means, and usually by competition that we are going to see in the next. So here is the overview of that, how the cellular repressors also control the SV40 transcription. So what we are looking at, that right here, we are looking at the viral late mRNA right here. So you can see the rise. But on the other hand, you can see that initially, if you just look at the time after the infection, initially the DNA replication is not happening. At this time right here, the early mRNA like is being produced, and the LT, which is the large T antigen, which is the regulatory protein, is being produced. Once they are transported back into the nucleus, you can see that there is an increase in the viral genome synthesis. And at that time, when the viral genome synthesis increases, that also enhances the late RNA expression, and how this actually happens along with the LT. So let's take a look. There is a protein, and these are the steroid thyroid family of proteins in the cell. These are called the IBP, or the initiator binding protein. And the level of this protein is pretty level. So over the period of time, you can see that this initiator binding protein right here, this level of the protein doesn't change over time. So what happens that when you have the genome of the SV40 coming inside the cell, these IBPs, or the initiator binding proteins, will bind to the regulatory region of the late transcriptional unit. So again, remember that they are binding to the late transcriptional unit, and they will not allow the transcription of the mRNAs. So what is now happening? So this is the basic process. So this is the genome, just a single genome. Think about that, just only one genome available. You have enough amount of this cellular IBP that blocks each and every transcription regulation unit, no transcription of the late gene. Now see that what happens. You have the increase in the genome, which is viral synthesis starts by the LT. Now you have large amount of the genome synthesized, and that will titrate out these IBPs. There is, as the amount is steady, it will titrate out the amount of these IBPs, not enough IBP to bind, so it will do the transcription of the late genes. And this is basically what is called the escape from the repression or anti-repression. The amount of increase of the viral genome actually does this process. So now let's take a look at a more complex, which is the adenovirus. And just remember that this happens in another several steps. So the first step, what happens, which we called as an immediate early, we have these names, immediate early, and this immediate early will produce this E1A protein that will go into the nucleus. That will stimulate the second round of mRNA synthesis, and this will produce the early mRNA synthesis, mRNAs, and then early proteins will be transported back into the nucleus. That will stimulate DNA replication, and that will also stimulate the third round of mRNA synthesis, which will produce the structural proteins to be assembled. So again, just I've written down in the notes in step by step. And again, just the overall concept, if you understand, it is very easy to remember. So first round of mRNA synthesis, second round of mRNA synthesis, third round of mRNA synthesis, and so on. And you can see that this is not important, but this is a virally associated RNA that we talked about. And this is actually, this is made by the RNA pol 3 in the nucleus. And this have some different function that we are going to talk about. And here, one of the most important one, that E1A protein control the transcription of the early genes, whereas this protein 4A and the L4, they control the expression of the late genes. That is the essence of the control. And that's what we are looking at here. So you can see that right here, the temporal arrangement of the adenovirus genome, and immediate early, early, and the late. So the immediate early will produce this E1A protein, which will stimulate this early gene expression and this production of this E2. And this E2 is required for the synthesis, DNA synthesis, and entry of into the late phase by the anti-repression or the relief of the promoter occlusion that we have just elaborated in the last slide. And then what will happen that in this phase also, it will produce these proteins 4A and L4. And the 4A and L4, although they are being produced in the late phase, they will enhance the expression of the late phase. So you can easily see that this is a cascade. This is a cascade. On the other hand, right here, this is an autoregulatory loop. So you can just easily, you can well imagine that how these are actually working in the transcriptional control. So that's the overview of the adenovirus. So now this is the question that we are going to ask my cell biology students. So here you actually see your famous retinoblastoma protein once again, and how that control the transcription. You know that the mutation of the retinoblastoma actually causes the cancer that was first discovered in the eye, and you have gone through the cell cycle and how it is. And see now that how adenovirus E1A exploits the RB so that it cannot arrest the cell cycle or the transcription anymore. So remember that RB is responsible for the cell cycle arrest because it doesn't allow the transcription. And this is how the adenovirus also used that. So what happens that this E2F, and this is a transcription factor, and along with that DP1 is called a dimerization partner. And when they are bound onto the viral DNA, and remember that this is what we are looking at is not the cellular DNA, although RB binds to the viral DNA, but it works the same way with the cellular transcriptional inhibitor, and RB binds to that. So what RB does, RB actually directly doesn't inhibit the function of the E2F, but RB actually recruits some of the specific protein, which we called as HDACs, or the histone deacetylases. So these histone deacetylases are responsible for tightening of the DNA, and they actually cannot transcribe the downstream of the gene. And again, this is, remember that we talked about that when the DNA is tightly wrapped around the histones, it cannot do that. And this is done by the RB, by the recruitment of the histone deacetylases. Now see that what the E1A protein of the adenovirus is doing, E1A just binds directly to the RB, so it prevents, so this is a competition. So it cannot bind to the transcription factor E2F or DP1. And at that time, it cannot recruit the HDACs and the E2F and the DP1. This is active, and the adenoviral transcription can go on. So this system was discovered, so the RBs was discovered in a retinoblastoma cell, but the process that how they can do that actually comes from our study of the adenovirus transcriptional control. So this is another way that the transcriptional control can be done by the cellular factors, biotranscriptional control. So here, again, you do not need anything. You just probably just thinking that this is crazy, but you can easily see that this is the double-stranded DNA of the adenovirus, and you can just appreciate that these are the transcriptional unit. Right here, you can see that this is your E1A right here, and this is your L4 that we talked about, and right here is the 4A2. So we talked about this L4, 4A2, and you can see that how diverse these proteins are in all over the place and how they are. So these are just an idea that how complex these controls are. So you have a single genome, but you have so many transcriptional control unit, and they are all regulated in different fashions that we just talked about. So the next, what we are going into, that the most complex herpes virus genome system, and this has a fairly large size of the DNA, and what it is doing so far, what we have seen right here, you can see that these are the cellular transcription factors with associated at least one host protein. Right here, this is just completely with the presence of just one host protein, and you can see that right here. This is the E2. This is the host protein. You can easily see that, and right here, one host protein. But here, what you are going to see that the virus is going to bring in a factor. So what happens? Let's take a look. It fuses, the herpes virus fuses, and it releases a protein which is called a VP16. So VP16 is immediately transported into the nucleus. On the other hand, the capsid core is transported to the nuclear pore, where the linear double-stranded genome is released, and immediately it is circularized. So what happens on top of this, you will have all your transcription machineries built in, but this protein, which is VP16, is absolutely necessary for this immediate early gene expression. So VP16 is absolute necessary. So that's the transcription factor that the virus is actually bringing in. So that's the unique thing that is of the herpes virus. So what it is doing in the first round, immediate early, the proteins, and this is easy to remember. The first round, they produce these alpha proteins. These alpha proteins are transported into the nucleus, and they actually help in the transcription of the second round, or the early genes, which produce the beta proteins. Now the beta proteins go back in there. It's the same thing. And they are responsible for the DNA genome formation. And the DNA genome of the herpes virus are actually produced in concatamers, and they are packaged and then cut off when the whole one size of the whole genome is packaged. That we could talk about later. But basically, they actually enhance the DNA replication. And the DNA replication, this template can again be used at the last part to transcribe the late mRNAs, which are going to make the gamma proteins. These are the structural proteins. Some of the structural proteins are synthesized on the ER, and you know that the ER proteins will be the membrane-bound proteins, and these membrane-bound proteins and these structural proteins will be assembled into the viral protein. So remember that these membrane-bound proteins are essential, which are the proteins on the spikes, or the spike and type of proteins, not spike type of proteins, which are present on the envelope. So you can see that alpha, beta, gamma, 3 different types of protein control the temporal regulation of the herpes virus. So here, the overview of that, the herpes virus brings the VP16, and the VP16 will immediately regulate the immediate early gene expression. And then the next 2 proteins which regulate is called ICP4 and ICP0. So ICP4 is absolutely necessary for the synthesis of the subsequent early and late genes as well. And ICP0 also enhance the early as well as late expression of genes as well. And but what happens, not expression, transcription of the late genes as well. And there are some of the replication proteins which are produced from the early phase that we have seen that enhance the DNA replication. And some of the mechanisms we also don't understand quite clearly. So you can see that right here, the beta proteins, which are in the purple, synthesis of that enhances the DNA replication. And that is being shown here. The replication protein, which are the beta proteins, enhance the replication of the DNA. So this is the herpes virus transcriptional program. So the next part that what we are going to talk about, and we will not going to, if we do not finish this, that is perfectly fine. That is not a problem. We have time. So is the viral processing. So now we have done with that transcription, and we're going to see that how we can make these mRNAs ribosome ready for the translation or expression into the proteins. So we just talked about this top line. And now in this process, we are going to talk about the other thing. The first thing that happens is that as the mRNA is being synthesized, a cap is put onto its 5' end. And this is done by essentially cellular RNA polymerase II, one of the subunit of that. And also there are, in some of the cases, there are viral enzymes responsible for that. Say, for an example, for the RNA viruses, there could be separate enzymes, or some of the viruses like VSV, the RNA-dependent RNA polymerase have all these capping function in the different domains as well. And actually, it's kind of funny that that capping function of the L protein was actually discovered by one of my colleagues, and we worked at the same time. His name is Tomoaki Ogino. He's at the Case Western Reserve University now, who actually discovered this capping function of the VSVL protein is at the same time. It was so it was published in Molecular Cell at that time. So very famous discovery for the first time, capping function of the viral RNA-dependent RNA polymerase. And anyway, so what is happening right here, what you are seeing, the cap structure is very unusual at the 5' end of the RNA. And this cap 1, you can see that this is the first base of the mRNA. And you can easily see that base 1, it could be usually this is A or G or A, and ribose sugar, and you have the triphosphates. But right here, you have a guanosine or G, which is, you can see that it is inverted. It is inverted, and it is connected by a 5' to 5' linkage. So you can see that here it is 1, 2, 3, 4. The ribose sugar is 1, 2, 3, 4, 5 here. Here it is the other way, 1, 2, 3, 4, 5. Here is the base right here. So the first reaction is that this addition of this guanosine, and this is done by the guanylyl transferase. And this guanylyl transferase can be associated with the RNA polymerase as a different subunit, a different enzyme in the eukaryotes that does the capping in the viral DNA, and it's known as the capping enzyme. It could be a different enzyme in many of the viral systems as well. The second thing that what happens to the cap after this addition of this guanosine at the 5' end, this residue at the 7th position is methylated. And this is done by an enzyme which is called as a guanine 7-methyltransferase in the eukaryotic system. And if there are viral guanine methyltransferases as well, or the part of the RNA polymerase as well. So essentially, it is the same process, but it can be done by different enzyme. But the process is basically the same. The second thing is that this is your cap1 structure. The cap2 structure is basically the first and the second base of the mRNA itself. And these bases are actually methylated at the 2nd position right here. So this is a 2' position, 2nd position of the carbon that is methylated. And we call this one as the O methylation. This is done by the 2O methyltransferases. So all these enzymes, guanylyl transferase, methyltransferase, 2O methyltransferases, are separate enzymes in the eukaryotic system which associate with the transcribing RNA polymerase and co-transcriptionally cap the mRNA. Co-transcriptionally, as it is transcribing, it will cap it. But sometimes in some of the viral system, it can do it post-transcriptionally as well. You can just have that. And some of the viruses will not have a cap, like for your polio virus will not have a cap. Use the iris. Remember, influenza virus will snatch the cap, so there is no formation of this kind of cap. It will just steal the cap from a eukaryotic mRNA and just synthesize the and just attach it to the first base. All right, so let's take a look at the second base actually. So let's take a look at the detail of this co-transcriptional capping. So what happens that what you were seeing, the start site, RNA polymerase incorporate about 20 to 30 nucleotides. And what it is doing that after it reaches 20, 30 nucleotides, you have a specific sequence on the transcription on the gene that is encountered by the RNA pol 2. And at that time, what happens that the C-terminal end of the RNA polymerase is phosphorylated. And this phosphorylation will cause some conformational changes, which will recruit the capping enzyme. And as the polymerase goes on, it will also cap the 5' end. So why this wait until the 20, 30 nucleotides? The reason being that what happens many times, if you remember that there are some initiator sequences, and without transcription factor, sometimes RNA polymerase may start synthesizing an mRNA, and sometimes it just falls off or abort the transcription after going a few base pair. On the other hand, the 20-30 base pair ensures that the proper transcription unit is transcribing, and this transcription unit is going to make a proper mRNA. And only at that time, once it makes these 20-30 nucleotides, where the system understands that it's going to make the mRNA, it's going to phosphorylate the C-terminal domain, and the capping enzyme will be recruited. So if the enzyme is making just 2, 5, 10 nucleotides and it will abort the mRNA, it's not going to spend its capping enzyme. And you know that in the nature, nothing is overutilized or something like that. And this is basically the conservation of the enzyme. All right. So after that, what we are looking at is the modification of the mRNA, and this is the cleavage and the polyadenylation of the mRNA. This happens in a very different way in some of the RNA viruses that we have seen, but what we are seeing in the eukaryotic system, as well as some of the viruses with the DNA in their life cycle, what they're going to do, that first, there are some very specific residues, and this is an AAU, AAA site, and followed by some GE residues, where there will be an endonucleotic cleavage of this. And once this is cleaved, this area will recruit several polyadenylation machinery, and then the polyadenylation will occur. Remember that in the pre-mRNA, the polyadenylation does not happen at the 3' end of the pre-RNA. Rather, inside the exon, there will be an endonuclease cleavage where you will have the poly-A tail added. And again, there are several different factors not needed, but the famous enzyme, which is actually needed, is the poly-A polymerase. And once this poly-A polymerase is recruited into this cleavage region, it will allow just a slow polymerization of this process. So see, this is the cleaved position. This is out, no function for that, will be degraded. Whereas right here is AAU along with the poly-A polymerase is recruited right here. It will cause slow polyadenylation. But after a couple of polyadenylation, about 10, 15 residues, another protein will be recruited, which we call as a poly-A binding protein 2, PAB2. Once that recruits, it will just set the poly-A polymerase on steroid, and it will rapidly synthesize about 200 to 250 residues of poly-A. And the why poly-A is necessary? The poly-A increases the stability of the mRNA and also helps in the export of the nuclear of the mRNA into the cytoplasm as well. So some of the viruses actually don't have, say for example, reovirus don't have a poly-A tail, and it is shown that some of the secondary structure of these reoviruses actually stabilize the RNAs without a poly-A tail. But it is essential for the stability of the mRNA. Otherwise, it will be degraded before it can synthesize the proteins using the ribosome. So we will stop right here, I guess. All right, so here what we are seeing that we actually stopped at this modification of the mRNA. And we were talking about the polyadenylation, as we have said, just to brush up or the material that we talked about, that there is a specific polyadenylation site, and you can see that this is very conserved, AAU, AAA, very conserved. Sometimes this U could be a G, but it is very conserved, followed by a GU residue. And then what happens that this is cleaved, and remember that this is not, we will see that this was the part of the pre-mRNA, and this is cleaved by an endonuclease. And after that, you can see that there are several factors that which is added, and these are all the cleavage factors that you can see that all these different names that you can see that overall you have this cleavage, polyadenylation, specificity protein, like those, and these all these steps and names are not required, but just understand that a large complex is associated with this specific residue, and the poly-A polymerase, this you need to remember, is also recruited. So once this is recruited, there is a cleavage of that one, and this will be the polyadenylation site. And with the help of ATP, there is a slow polyadenylation that actually happens. And you can see right here, the poly-A polymerase, which is right here, this actually remains, but all other CF1, CF2, CSF, they are all dissociating. What we mean by that, their function was all for the cleavage of that. Once the cleavage is done, they are gone, and the poly-A polymerase is staying in there. At this time, the poly-A polymerase do polymerize about 10-15 nucleotides, but it is really slow. So what it does, that there is another protein, which is the poly-A binding protein 2 or PAB2. This also, please remember that. So just only two, poly-A polymerase and poly-A binding protein. When poly-A binding protein comes in and binds in here, as I said before, the poly-A polymerase actually just goes on steroid, and it just rapidly synthesizes about 200 to even 250 A's are added rapidly. And that makes the mature mRNA to some extent. This is one part of the mRNA. And along with that, what we are going to see that there will be some splicing mechanism. And the thing is that also we are talking about that all of these phenomena that we are seeing, that polyadenylation, then splicing that we are going to see, the capping, we have separated these out, but these are all actually, most of them are happening as the transcription is happening. So this is co-transcriptionally, most of these actually happen at the same time. And the interesting part is that the C-terminal end of the RNA polymerase is actually a very important factor, and these actually carry out almost recruitment of all of these in there. And we talked about that right here. And you can see right here is the RNA polymerase, and right here the C-terminal phosphorylation actually recruits the capping enzyme, and it is responsible for recruiting many of these functions as well. So it happens, all of them happen actually co-transcriptionally at the same time. Very quick process. So after that, we are taking a look at how the poly-A tails are added to the viral mRNAs. So one process, which is like the eukaryotic system, you can see that this is post-transcriptional. So in the post-transcriptional process, the cleavage of the mRNA is followed by the polyadenylation. And again, remember that that does not mean that they are all separated out, but this is after the cleavage of this specific polyadenylation site. That means it is post-transcriptional in that sense, although they are happening at the same time, but it has to be transcribed first with the sequence, which is the cleavable sequence, before the polyadenylation is added. That's what it is. So the enzyme is cellular, poly-A polymerase, and the associated enzymes, and you can see that many of these viruses that we are going to talk about, adenovirus, herpes virus, polyoma virus like SV40, they actually do have, including retroviruses, use the cellular machinery. Whereas you can easily see that this is the completely co-translational, means that as the mRNA is being synthesized, it does not produce any cleavable site. So this doesn't happen. So one of the thing is that the reiterative copies of stretches of U of the template RNA, and you can see here that for the influenza virus, as we have seen, and the VSV, both are RNA viruses. And there is a stretch of usually 7 U's, or sometimes about a little bit different number depending upon the virus. And the RNA-dependent RNA polymerase do stutter or slip on those U residues and pump out these 150, 200 polyase after that. So very different from here, very different enzyme polymerase. Here the RNA-dependent RNA polymerase does add those. And that's the reason that we are talking about the mRNA synthesis happens, or the enzyme for these RNA viruses are the RDRPs, or the RNA-dependent RNA polymerase. They by themselves add these poly-A tails. On the other hand, here is another, this is copying a long stretch of template RNA. In poliovirus, actually, they have a considerable long stretch of RNA, not a short stretch of RNA, long stretch of RNA. Poliovirus just make those copies in that. So the poliovirus is plus strand. So the plus strand poliovirus is already polyadenylated. So when they make the minus strand, they will make these long U's, and on the U's, you will make the A's. So that's basically the overview of the process in the poliovirus. In coronavirus, this process is a little bit difficult, and I just put a question mark in there because we do not understand this process completely, although there's a couple of papers. You can see that even a paper came out in 2019 suggesting some of them. And in the coronavirus, the number of polyase which are added actually do varies. And there are some evidence that the same way that slippage of the RDRP actually happens, but there is also evidence that along with the RDRP, there is an additional enzyme which is known as the protein B is also responsible for adding the poly-A tail. And there is a very specific polyadenylation signal in coronaviruses, very similar to the eukaryotic polyadenylation signal. And again, you do not have to go into that detail of that at all. We are not going to ask that. This is just for your interest because we are just all talking about the coronaviruses. So it's a much more sophisticated and complicated procedure, which is not completely understood yet. All right, so after polyadenylation, what we are looking at is the next process, which is splicing, the famous splicing. So how it actually happened that this was discovered in adenovirus infected cells. So you can easily see the cell biology or molecular biology all actually, as I say, stemmed out of these virology research in there. So what the scientists actually saw that when they isolated the mRNAs from the adenovirus infected cells, they actually see what is called a heterogeneous population of nuclear RNAs. And these heterogeneous population, the mRNAs are of all different lengths. And they could not actually understand that why there is so many different length of RNAs. And they were just puzzled with that, very different lengths. And so what they did, that they actually took a specific adenovirus mRNA, and mainly the hexon mRNA, that was the original experiment. And this is the mRNA which produced the protein. And they hybridized them with some of these heterogeneous nuclear RNAs, and they were labeled by some of the electron-dense material, and they looked it under the electron microscope. And what they saw, that just like this kind of a picture, they saw these huge loops are coming out from the RNAs, whereas the other one, the mRNA, is hybridized on the base of it. So this is the basic understanding of this. So this is, you can easily see that this is how it was discovered. And then Richard Roberts and the Philip Sharp actually said that, or proposed the thing, that there are some of the untranslated regions which they call as an intron, and these are spliced out to form the mature mRNA. And you can see that how it is that you have these tripartite leader sequences, and these are all in there, and you can see that loops, these are the loops which are right here. And that's basically the overview of that, that how it happens. So you can easily see that this is the hexon mRNA body, which is this, and these are just the loops right here, which are not present in the matured mRNA. And that gave the first proof of that splicing is actually a phenomenon. And a process that immediately within 6 months and 1 year, this splicing has been seen in all different cells, and almost is an universal rule in the eukaryotic system. Prokaryotic system, it doesn't happen. So it's a rule in the eukaryotic system, and lots of system has been identified after that. And so very famous discovery. So what is the process that how these pieces and why we have this splicing mechanism? There has to be a reason for that. So first, let's take a look at the process by how the pre-mRNA is spliced. So if you just take a stretch of the mRNA, what you are seeing that the green part is an exon, which will be in the part of the mature mRNA. This is the exon 2. This will be in the part of the mRNA, whereas right here in the middle is the intron, and this will be spliced out so that exon 1 will be joined to exon 2. This is basically the splicing process. So now if you look at the bases right here, you can easily see that right here you have a residue which is GU, and these GUs is right here is the frequency on occurrence in hundreds and thousands of different individual mRNA. And you can see that these two residues are 100% conserved. What it means that you just take any organism, isolate the mRNA, and look for these sequence as a 5' splice site, you will have those GUs. On the other hand, look at this A, this is also 100% conserved. On the other hand, if you just look at this A right here, these AGs, these are also 100% conserved, and these are the 3' splice site. So you can easily see that where the cutting is happening, those residues are extremely conserved throughout all the different ones, and the viral mRNAs are also no exception. It is the same thing for them as well. So let's take a look at how this splicing happens. So what you are seeing right here, you have the exon 1 followed by, right here, it is just like a loop, these GUs. So you have the GU. So the G right here, exon 1 and the 5' splice site, you have the phosphate. And we're going to see by a specific mechanism, these two strands comes very close together. And this right here, this A residue, which is very important, this actually attacks, the hydroxyl end of this actually attacks the phosphate, and this is a transesterification reaction. So this phosphodiester bond is actually cleaved, and here you can see the 5' exon is out, and this is right here is done by the transesterification, by this. Now what happens, that once this is actually gone, you can see that this is now joined together, this G and the EU. Now what happens, you can see that the 5' end of this exon 1 has an open 3-hydroxyl end. And this hydroxyl will now again attack this phosphate of this exon's 5' end, which is the 3' splice site of the whole mRNA. It's a 5' end of the exon, but this is the 3' splice site. Just remember that. So this 5' splice site, 3' splice site. So once this is attacks this phosphate, this will be again transesterified, and you can see by that process, this lariat-like structure is excised out, and these two, the exon 1 and the exon 2 is now joined by the phosphodiester bond. And this is a process which is extremely complicated, and it is done by a number of proteins, as many as 150 proteins that has been discovered, and RNA complexes. And we are going to see that how this actually happens. So here you have a very complicated diagram, but again, you do not have to remember all of them at all. If you just get the overview of that, that is good enough. So here you can see a pre-mRNA with just a hypothetical one, and you have this right here, this intron. And around this intron, you can see that there are a number of proteins which actually associate. And along with that, so the proteins are just made with these round structures, but there are lots of these, which we called as the nuclear RNAs. And these nuclear RNAs actually associate with these area, and they are named as like U1, U2, U4, then U6. So you have all of these U5, so you just only don't have U3 in here. So other than U3, there are 5 of them, U1 through U6, except the U3. So all these actually assemble around a structure which we called as a spliceosome. So the spliceosome is nothing but basically a ribozyme, which is basically a catalytic RNA. So you can easily see right here, this catalysis is done by an RNA. So the part of the RNA is attacking the part and doing this reaction. So basically, this is we always have seen that these reactions are carried about by enzymes, which are proteins. Here, the catalysis is done by the RNA itself and the ribozyme. And this is probably the relic of the RNA world, where the RNAs actually evolved this way by their own catalysis. So this is basically the spliceosome is nothing but a ribozyme. So what else is from here? So you can easily see that right here is the GU residue, which is important, and that is the AG residue. What these RNAs are actually doing, these small nuclear RNAs are bringing these specific residues close enough so that these transesterification can happen. That is the only take-home message in here. That is the main purpose of these different bond formation, just so that these catalytic sites of the RNAs are brought close. And you can see that easily that how the splicing out is happening, and the whole process in here that we have seen before. It is just the breaking and making of these different hydrogen bonds in between these small nuclear RNAs to and the intron and the exon of the mRNA, just to bring the splice site close together for the catalysis. So this is basically the overview of that. So that's all you need to know for the whole purpose. That's just an overview. In individual steps, what is happening is not at all required. So what this gives it to you. So due to this, there are 2 things that can happen. One is great, okay, you have a constitutive splicing. You have an exon 1, exon 2, exon 3, and you can have joined them 1, 2, and 3. So these have implications as well, because in the eukaryotes, many times what happens that recombination can happen, and a recombination can happen in the DNA. Just say for the human being, during the meiosis, you know that the recombination actually happens during the crossing over, and sometimes if the recombination happens in here, there are chances of a loss of information from the real exons. Actually is a possibility. If you have a crossing over, sometimes these areas which are sensitive can break, and the protein sequence can mess up. So the recombination can happen into these exon areas, keeping the coding sequence intact. That's constitutive splicing, and we have seen that all the time. On the other hand, what you are seeing that this is actually used by the viruses all the time, but again seen in many of the eukaryotes, particularly for making some of the proteins with extreme variations as well. We have seen this as well. Say, for example, antibody production, we will see that. So here what you are seeing, that the alternative splicing, you can easily see, very self-explanatory. You can join 1, 2, and 3 all together, or skip the 2 and join 1, 1, and 3. So that's exon skipping. On the other hand, you can see that right here, you can have a 2 splice site, a 5' A and a 5' B splice site. So you can easily see that 1 and 2 can join, and then 1, along with this sequence of the exon, can join with 2. So you have alternative splice site. On the other hand, here you can see that you have an alternative 3' splice site. So at the 3' end, you have this. So this is a 5' variation in the splice site, 3' end in the variation in the splice site, give you different types and organization or outcome of the mRNA. So that's basically what we see in many of these viruses. And let's take a look at this, where you can see in adenovirus, this process is at its epitome. So how elegant this system is, and we will see that this is, you have a specific length of your DNA, and you can make a huge amount of mRNAs at different times in different quantities and in different shapes and sizes, and that actually gives you a huge output from the genome. So let's take a look at what is happening in here. It is not as complicated as it looks like from this. So you can see from the genome right here, from the major late promoter, you have this mRNA synthesized, which is a 5' cap, and then you have some right here, this 1, 2, and 3, these what we called as these leader sites right here, very small exons right here. And then you have an L1, L2, L5. These are the other exons that you can see. But what you are seeing, that your L1, 2, 3, 4, 5, these are all polyadenylation sites. So that means that you need to cleave this out and then put poly A tail, cleave this one at poly A tail. That can happen. So in the early phase, when the adenovirus is replicating, what happens that the cleavage only happens in here, and this L1 transcript is only formed. Only the L1 transcript is formed. And then as the infection goes into the later phases, all other mRNAs are produced as well. So these are now the pre-mRNAs that has been produced at different times. So now we are looking at how these are going to be processed. Now remember that. So the purpose of this is polyadenylation. So here you add the poly A tails here. This is first made, then poly A tails here, poly A tails here, poly A tails here. These are already added. They are not spliced yet. So they will be spliced after this process. But again, as I repeat what I said, that at the initial phase of the transcription, this one, which is the L1, is made in the largest amount, and then pretty much all of them in equal amount. So now here is a very interesting thing happens. You can see that by the alternative splicing, this 1, 2, 3, these 3 regions remain, and then you have added the L1 splice site to that, and you have an alternative splice site. So from the L1, you can get 2 mRNAs, and both of them will have the initial leader sequence of these 3 exons. So these 3 dots are these 3. You have it in here in the L1 mRNA, which is a little bit more. This is a 52, 55 kilodalton protein, and the protein 3A. Both of them have the same initial sequence, but they are different proteins. On the other hand, what you are seeing right here, there is another way that there are 3' splice sites possible, 4 possible splice site, where also you have these 5' leader sequence, just the same way that you have. So all of these mRNAs that are arising from this alternative splicing will have this tripartite sequence of this 1, 2, and 3. And right here, you can see you can have an L1, then you can have an L2, and by this alternative splice site, you have a large number of L3 mRNAs that can actually be produced. So you can easily see that this is protein 4, hexon proteases, and many more. This is just a few of them has been shown. And all these mRNAs or proteins can arise from this alternative splicing, this elegant mechanism. So what is, so basically that's the outcome, and we are going to see at the end that what is basically the reason for that. The reason for this is nothing but the efficiency of the coding of the genome. A small genome can make a large amount of mRNAs and many different proteins. That's basically the overview of the reason for that. Also, along with that, the splicing also marks these mRNAs to be exported from the nucleus into the cytoplasm, which is where the ribosomes are for the translation. So the next one, what we are seeing right here, is the splicing marks the mRNAs for the nuclear export. So what you are seeing right here, and again, there are lots of different names and proteins, and all of them are not required. Just an overview of the process, if you just understand, that's great. So what you are seeing that right here is the exon, then you have this intron marked like this hashes, and then you have the exon 2. And here you can see that this is the ribonucleoprotein complex, heteronucleus ribonucleoprotein, and these are the spliceosomes. And along with these SRs, right, these are all the proteins which are required for the splicing. They all assemble together. And there is a cap binding protein, several different proteins, all assemble. So you can easily see, as soon as this polyadenylation is done, a large number of protein are associated on these mRNAs. And after splicing, you can see that all these proteins come together, and then these proteins actually do recruit something which is known as NXF1. Now, this is a misprint in the, I need to kind of like I did not have time, but I have wrote that NFX1 should be read as NXF1, because NFX1 is a very entirely different thing. It's a new, it's a transcription repressor. So I mean, like, if it is just a typo, then it is fine. But if it is NFX1, you put it in there. There is something which is called NFX1, which is very different from NXF1. So what NXF1 is, and remember only that name, which is the NXF1, this is the nuclear export factor. So basically what we call this, that these are like adapter proteins, and when they attach to the mRNAs, they can be transported through the nuclear pore through the cytoplasm. So the take-home message is that after the splicing, a number of protein actually assemble on the splice site and on the mRNA, and then the nuclear export factors assemble on them, and that marks that mRNA to be transported into the cytoplasm. So that's basically the overall process. So now the viral RNAs will be transported this way as well, but there are some variations in there. Remember that one of the very important thing is that for retroviruses, there is a problem. Retrovirus does have a DNA, and which is integrated into the some of the chromosome that will make these mRNA. But remember that for the retrovirus, plus sense RNA is the viral genome, and that has to be unspliced all the way from the 5' end to the 3' end without the loss of any nucleotides. And how do you transport those? And this happens in all the different retroviruses. And so there should be a mechanism that the unspliced RNAs can be transported out of the nucleus as well. So that's what we're going to see next. So here is the same picture that you have from the previous slide, the export of the cellular mRNAs. Here you have an unspliced retroviral RNA. And remember that this is a retrovirus with a simple genome. And I just mentioned that clearly. in the note, remember that this is not an example of HIV, which is a retrovirus, which is a much more complex genome. This is a genome of a very simple retrovirus. And what they have, that they do have a intrinsic right here element or intrinsic sequence, which is a constitutive transport element. And these CTE is the site which is recognized by this NXF1, and also there is another nuclear export, NXT1, and these are all the transport adapters. So what you are seeing, that the full length of the RNA already contain a constitutive transport element. So even if they have this right here, the intron doesn't matter, the adapter protein will bind. In here, the adapter proteins will only bind if they actually just go away. So you can see that this is dimerization of these SR domains are required. So one of these SR binds to this 3' splice site, one of them binds to the 5' splice site, and they can only be together once this piece is gone. And this, when they come together, that will recruit the NXF1. But here, it is not even needed in the recruitment of any of these elements. Directly, there is a sequence which is recognized by NXF1, and that will actually transport them through the nuclear pore. And the reps are some of the associated proteins as well. So this is for the unspliced retroviral mRNA with the simple genome. Whereas in HIV, their system is way more elegant. So what is happening right here? So think about that. That's your integrated genome of the HIV genome, and you have the transcription, and you got the whole length of this long mRNA, and you will have also the minus RNA intermediate, but you will have the mRNA done from that portion is at the very beginning. So when the cell is infected by the HIV, that is a separate process where the reverse transcriptase will make a minus RNA. From there, it will make the DNA that will be integrated. Then that whole portion is gone. That reverse transcription is not happening again. Here you have the integrated DNA and the RNA polymerase, cellular RNA polymerase, are going to make the mRNAs. So now the first thing what happens, that once this is done, there is a secondary structure, and this is known as this RRE, or the Rev response element. So what is Rev? What we are going to see, that once this long mRNA is produced, this is actually spliced, and a small mRNA is transported into the cytoplasm, small mRNA, and this small mRNA is going to synthesize the Rev by the translation. So this Rev, this protein, which is from a spliced mRNA, goes back to the nucleus. And once Rev goes back to the nucleus, the Rev will bind to this Rev response element right here, which is formed by the secondary structure of the full-length mRNA, which is the viral genome itself. Once that happens, this interaction of the Rev, that can transport the full viral genome into the mRNA. Sometimes some of the other long mRNAs, which needed to be transported in form of partially spliced. So you can have many spliced region, but you do not want some of the spliced region to be spliced. And those are also transported into the cytoplasm by the Rev. So you can easily see that right here, because all the splice sites are spliced, that's the reason that the small Rev, this mRNA is exported into the cytoplasm, Rev is produced. But these large ones cannot be transported. These are all transported once the Rev goes back in there and transport them out. So so basically what will happen, the Rev is recognized by a transportation adapter, which will bring in. So as the Rev is transported, remember that also, as Rev is transporting this mRNA, Rev will also come out in the cytoplasm, and it can be cycled back again to bind to another mRNA to bring them back again. So this is how either the full length or large partially unspliced mRNAs can be transported in the cytoplasm by the HIV. Very, very unique process. So what is this all about? Well, the splicing creates what we call as a value-added genomic expression. It creates different mRNAs, proteins, coding information. This is the most important information. Coding information of a small DNA genome is actually expanded. And along with it also regulate the gene expression. What we mean by this, that as these things are transported in a temporal manner, initially you synthesize some of the specific proteins, which we called about from the L1 mRNA, and as we go into the late phase, the other mRNAs are produced to make proteins, particularly for the structural proteins. So those are the things that regulation of gene expression, so that the gene expression is controlled in a temporal manner. Not all the proteins are produced at the same time. Initial regulatory proteins are produced in the immediate early and the early phase, and then the structural proteins are produced towards the late phase. So that's pretty much it.